Hello and welcome to video lesson 4 of our DNA unit. In this lesson we will be talking about the cell cycle. The cell cycle. So what is it? Why are we talking about it? Well, to start with, all living things have a linear life. What that means is, that is not a B. We are born, we live our lives, and then we die. Linear, okay? We're born, we live a great life, hopefully, and then we pass away. Cells' lives, however, are circular and cycle in nature, and it's a repeating pattern. So the repeating pattern of events in a cell's life is called the cell cycle. So our lives, we born, we die. Hopefully in here we have reproduction, so we'll have sons and daughters. But a cell continues to have this cycle, this life cycle, very circular in nature. There are 4 major parts to the cell cycle. We have G1, S, G2, and M. And we'll go into these in great depth in the next few slides. This is a diagram of the cell cycle, and you can see here's our G1 phase, our S phase, our G2 phase, and our M phase. Okay? G1, S, and G2 all belong in a phase called interphase. Okay? So if you look, this is all interphase. G1, S, and G2. And then we have the mitotic phase, and the difference is G1 simply stands for growth. S is the phase for synthesis, and what gets recreated here, that is DNA. So we have growth, DNA makes a copy of itself or synthesizes, and then we have G2. What do you think happens in G2? Hopefully you said another growth phase. So our cell grows, it copies its DNA, it grows again, it then moves the duplicate copies of stuff into 2 separate cells, and the process of cytokinesis cuts the cells apart, and we had 1 cell turning into 2. That is the cell cycle. So let's get into this. If you guys like words more than pictures, G1, again this phase, G1 is growth 1. So it's the time when the cell does its job. DNA is unduplicated, so whatever it needs to do is producing energy, it's acting as a skin cell, whatever it needs to do, that's what happens during G1. Synthesis, or the S phase, is where DNA is duplicated or copied in the preparation for the cell to split. DNA is now has duplicated chromosomes, and again, that's the S phase. So we grow, life's happy, we then synthesize or make a copy of DNA. We're then going to grow again. In this phase, our cell goes through a process of proofreading to make sure that the new DNA we created in the S phase, this is now the G2 phase we're in, make sure that the DNA is correct or is correctly duplicated. Once that proofreading ends, we then enter the mitotic phase or mitosis, otherwise known as the M phase. And in this process, the cell splits into 2 new daughter cells. Each one is absolutely identical to the first one, and then the cell cycle begins again. Okay? So chromosomes look like this during G1 phase, or before the S phase. So here's our chromosomes. You can see these color bands here, there and there and there. These color bands just represent where specific genes would fall on a chromosome. So for example, this could be a gene for eye color. This could be the length your fingernails grow. This could be the pigment of your skin. This could be your hair shape, whether it's wavy or straight or whatever. Okay, so different genes. Chromosomes before we duplicate, before the S phase, during G1. We go through the S phase, and during G2, we have what are called sister chromatids, so identical copies of our original DNA strand. Okay, it is a duplicated copy. So before the S phase during G1, DNA is going to condense into chromosomes. We go through the S phase where we get that duplication, and then after the S phase, we're ready to split these two apart into two new cells. Okay, so when we have duplicated DNA in G2, so after the S phase, it is called a sister chromatid. So the cell cycle does regulate itself, and there are checkpoints to make sure that happens. So proteins are there as checkpoints, as like the little police officers or guards to make sure stuff is happening correctly. And they decide if a cell can proceed from one phase to the next. So from either G1, S, G2, and M, you can see there's different checkpoints along. Okay? The G0 phase, or cellular senescence phase, at that area, the cell will either stay in G1 permanently, and then cell no longer grows or divides. So when a cell has reached its maximum output, it can enter into this phase. Here we are. Enter into this area to where we no longer go through cell cycle. It just stays permanently in G1, its normal state. Apoptosis means programmed cell death. The cell self-destruct when there is too much damage to fix. So here it goes through the cell normal life cycle where it reproduces. It then enters GO, or it stays in G1 permanently until we get to the point to where so much is wrong with the cell that something is triggered inside of the cell and we get apoptosis or cell death. And remember, this is part of that normal body process to have cells die, and that eventually will leave your body or get recycled into other parts. Our Geek of the Week is Henrietta Lacks. She died of cervical cancer in 1951 at the young age of 31, and she had one of the most aggressive cancers ever reported. Her cells were taken from her during surgery, and specifically, notably, without her consent, and it was used to create the HeLa cell line. This HeLa cell line has been used to study and cure many, many diseases, everything from polio and helping to get us a cure on AIDS. Okay? So it helps us study many, many diseases that are out there. And remember, she gave her cells without any form of medical consent. So is it worth the price of immortality? She's done a whole lot of good for millions and millions and millions of years, or millions of lives, I should say, but she did it unwittingly. She didn't have, her cells outlived her. And the question is it worth it? And that, my friends, brings us to the end of this video lesson on the cell cycle. As always, thank you so much for your attention, and I look forward to seeing you in class soon.